assembly. And here, the interesting thing is that we have not given any additional stress to these neurons, right? We have not given any oxidative stress. So, meaning that the mutation by itself, the mutation of this protein by itself acts as a chronic stress to these neurons, right? The R521G and the P525L mutations, in both cases, it acts as chronic stress and causing formation of these FUS and G3BP aggregates in stress granules. And as proof of concept, when we targeted this pathway, the PERK pathway, PERK is an enzyme uh, which is involved in the phosphorylation of the EAF2 alpha protein, which I showed you in the beginning. So that is the one of the initial pathways wherein the stress granule formation starts, right? So we asked if we can prevent the phosphorylation of this EAF2 alpha protein, can that prevent the stress granules from forming? And the answer is yes. As you can see here, both FUS and G3BP, the stress granule protein, they do not undergo this accumulation into stress granules anymore. And here you can appreciate with the drug, the PERK kinase inhibitor drug, the number of neurons experiencing these stress granules is drastically reduced compared to the non-drug treated ones. Then we asked, so what happens? Why is this stress granule pathway anyways important, right? So what will happen if there is a chronic stress event? So to understand that, we carried out some time-lapse microscopy, wherein we imaged the same neurons for 24 hours. And what we identified is that in case of these mutant cells, so this is 0 hours and this is 24 hours. What you can appreciate is that the same neuron after 24 hours undergoes cell death, which is shown by the accumulation of this dye called RAC7. So whenever the nucleus of the neuron is compromised, this dye can enter the nucleus and it can act as a marker of cell death. So when we treated neurons with the PERK kinase inhibitor, this is prevented, meaning that initially there is no stress granule formation even after 24 hours, and the cell is completely healthy. And when we quantified this over a period of 5 days, what we can see is that in the presence of the mutants, the P525L mutant and the R521G first mutation, the cell survival is drastically affected, meaning like almost 50% of the, around 50 to 60% of the neurons are killed. And this is restored in the presence of the PERK kinase inhibitor. So again, emphasizing that this phosphorylated EF2 alpha integrated stress response pathway is very important in causing toxicity through FUS mutation. And the other interesting thing is that when these mutant FUS stress granules are formed, what happens to the biophysical property of this protein, right? So to do that, to understand that, we carried out a technique called fluorescence recovery after photobleaching, or FRAP, in which you kind of bleach these regions of the aggregates, right? So this aggregate is bleached and we allow some time for this to recover. And then we ask how much of this initial fluorescence gets recovered, right? So this is mostly dependent on how mobile a protein is. If a protein is quite mobile, then it means that the recovery of this fluorescence will be faster, right? And what we observe is that without any drug treatment, there is a very slow recovery of the fluorescence in case of this mutant P525L protein. But when you add this PERK kinase inhibitor, this recovery is fast, meaning that we are able to also change the mobility of the protein using this PERK kinase inhibitor. So essentially, because the protein is in an immobile state, it is causing what is called the called as the traffic jams. And if we use this kind of approach wherein we are targeting this PERK kinase, it can prevent that protein aggregation and the traffic jam like situation. So we have also calculated the mobile fraction here. Essentially, it shows that in case of the PERK kinase treated ones, the mobile fraction is more compared to the non-treated ones. And inversely, the half-life of the protein is reduced, meaning that the turnover of the protein is faster in case of the PERK kinase treated ones. So to summarize this part of the talk, so what I have shown is how this mutant first can affect motor neurons in ALS is that when you express mutant form of FUS, it causes ER stress by phosphorylating this protein called AF2 alpha and causing chronic stress granule formation. So these chronic stress granules are highly stable in nature, and the cell can not handle them by either proteasomal degradation or autophagy. And this then leads to translational repression, meaning that a lot of essential proteins are stopped from translating, right? So, and as I shown in the beginning, like neuronal activity is dependent on how fast synaptic neurotransmitters are released, right? So for neurotransmitter release, we also need synaptic vesicles to be synthesized and transported. So because of this traffic jam kind of situation, these synaptic vesicles get trapped also in these stress granules, and ultimately this leads to neuronal death. And by targeting this PERK kinase pathway, we are able to rescue all these things. And for the second part and final part of the talk, so the interesting question is what determines the age of onset and severity of the disease ALS, first ALS. Different patients come to the clinic at different ages. So few patients are as early as 40 years and few patients are in their late 60s. So why there is this kind of discrepancy in the age of onset? And also some patients who have the disease, they die quite fast, maybe in the first 1 or 2 years, and few patients survive more than that. So what is the reason for this? And can we target pathways to prevent this kind of neurodegeneration, right? So towards that, we have identified things called as genetic modifiers that can regulate these stress granule dynamics. So highlighted here are two studies wherein we have shown that there are genes other than FUS which can also regulate the ability of FUS to form these stress granules. So there are two different genetic modifiers we have identified. One is called PUR alpha. This is again the RNA binding protein. So what essentially we found is that when you overexpress this gene, it kind of enhances the turnover of stress granules, and also the FUS no longer forms these aggregates. And the other recent study is on a gene called muscle blind. So this is a interesting study along with our collaborator, Dr. Uday Pandey. So wherein this gene is typically implicated in another disease called myotonic dystrophy, which has no relationship to ALS. So initially, it was kind of surprising that knocking down this gene called muscle blind was protecting the motor neurons from FUS toxicity. So how did we first arrive at this gene is that we did like a genetic screen in Drosophila, wherein we expressed a lot of overexpressed a lot of genes and knocked down many genes, and then we came to the conclusion that by inhibiting this gene called muscle blind using a RNAi approach, you can preserve the eye phenotype. So shown here is the eye of the Drosophila fly. It's a fruit fly. So shown here is the microscopic examination of the eye phenotype. So if you have something like this smooth structure, it means that the eye neurons are intact and they haven't died. Uh, when you overexpress muscle blind, what you can observe is that in case of both the wild type and the mutant, what you can see is that there is a complete loss of these neurons in the eye. And when you use a RNAi approach, you can certainly rescue this kind of eye degeneration phenotype, which is quantified here, underlying that. So this gene indirectly regulates first toxicity. And so it's a long paper, but for this talk, I'm just summarizing the very important point. You are welcome to go through our publication, recent publication. So how FUS is able to interact with the muscle blind at the genetic level is that so FUS enhances accumulation of a critical axonal protein called SMN. So this is a shown in green here. So this is the axonal protein, meaning that this is involved in neuronal communication, as I showed in the before. So this protein called SMN, or survival motor neuron protein, it gets trapped in these stress granules, right? So in the presence of muscle blind, this gets trapped into these aggregate structures. The green gets aggregated into these FUS and forms these stress granule structures. And when we use a RNA approach, shRNA approach to knock down muscle blind, then what we observe is that this SMN is now seen in the axons as it should be in physiology. So muscle blind is indirectly regulating the function of this SMN protein. So therefore, whenever we knock down muscle blind, it can prevent the accumulation of SMN into stress granules. So thereby, we are rescuing some of the neuronal function. And also here, using time-lapse microscopy, we have evaluated if this muscle blind gene causes neuronal death by using this shRNA approach, wherein you can see that when we overexpress the mutant form of the protein here in red, along with shRNA labeled in green, you can still see that the neuron survives for 72 hours. But in the case of scrambled shRNA, the neuron already starts dying at 48 hours. So again, this is quantified here, suggesting that overexpression of muscle blind leads to neuronal death caused by FUS. So to summarize my talk, what I have shown you today is that in a healthy neuron, the nuclear FUS is intact. It's localized mostly in the nucleus. And what happens with disease is that there are a lot of triggering factors. One is aging, and the other one is cellular stress. Like a person can undergo viral infection, so that can also act as a cellular stress. And the one I have showed you today is disease mutation, right? So this disease mutation can trigger formation of these cytoplasmic FUS aggregates, which then get trapped into structures called stress granules. And these stress granules prevent protein translation and transport. And some of the critical synaptic proteins are reduced in case of ALS FUS neurons. And this in turn prevents the synaptic vesicles or neurotransmitters from releasing properly. And carrying forward this work, so we are also in the process of developing brain organoids, which are kind of mini brains or mini spinal cords by themselves, right? So all the studies I have shown you before are like mostly isolated structures, mostly neurons by themselves. So the question now comes, like, how do we model this in a organism level, right? So this is a very closest model. wherein you can build a brain or spinal cord from like a small neural ectodermal stage and all the way to a brain-like structure. So this brain-like structure expresses different cell types like neurons, astrocytes, and initially they are in the stem cell-like stage, but slowly as we age them for like 60 days, we are able to find these other cell types there, neurons and glia. So the challenge now is to model this kind of first ALS using these brain organoids. And with that, I would like to thank my lab members and collaborators for their support. And thank you for your attention. And I'll be happy to take any questions.